In this video, we will discuss about some of the strategies by which we can modulate the circular RNA in the mammalian cells. So, first strategy is about the upregulation of the circular RNA through different ways or through different methods. First one of them is the intronic repeat mediated back splicing, or in simpler term we can call it as co-transcriptional back splicing. So back splicing will occur at the time of transcription, and this is how it will happen. That is, this pre-mRNA is translating. At the same time, there is a bond formation occurs between two parts of the pre-mRNA through this spliceosome. And the next approach is to use a tRNA intron in order to circularize the RNA after it is transcribed, or this is known as post-transcription strategy. It is, we will use a pre-tRNA to ligase both the ends of this mRNA that is transcribed from the DNA sequence. The third approach is the permuted intron exon splicing. So what happens is that there is a self splicing that is occurred. Self splicing group is incorporated into the mRNA in such a way that there is a splicing occurs, and that some of the splicing might result in circular RNA, however some of them does not result in circular RNA. It depends on the permutation and combination of which intron is spliced and which intron is not able to splice in a particular transcribed RNA. Next one is the induction of the exon splicing. So in this, what happens is that when the pre-mRNA is formed or linearized, and during splicing some of the intron is further removed through this intralayer back splicing. However, this strategy is right now not been proved, and this is only right now only a theoretical concept. So in this, what they use DCAS13. That will actually does is circularize first this two flanking part of the intron, and then this intralayer back splicing will remove the intron part and helps the exon parts to come together and make a circular mRNA. These are some of the strategies for circular mRNA upregulation within the cells or in the mammalian cells. Next one, we will talk about the depletion, how we can deplete the circular RNA. First one is to use this RNAi, or you can say short interference RNA or RISC in the sequence, or transfected them into the cells so that it can cleave off the circular RNA at the specific sites for which this SIH RNA is designed. Short interference RNA. So next one is to use this RNase H instead of this siRNA. It can do the same function that is done by this RNAi. And third one is to site specific depletion, or to deplete specific kind of RNA only, not a group of RNA. In this, we can use this CRISPR-Cas13 and the CRISPR-Cas13 system, and design the guide RNA to cleave off a specific site of the circular RNA. And another thing that we can do is to use this ASO, that is antisense oligonucleotide, so that it can target only the circular RNA and whenever this CRISPR-Cas system tries to target the linear RNA, since the antisense oligo is bind to that guide RNA sequence, so that now this CRISPR-Cas13 system will not be able to detect or attach to the site of the linear RNA. In this case, it can only clipped off the circular RNA. However, this antisense oligo need to be selected in a way that it can only bind to the linear RNA, not the circular RNA. At the same time, it does not affect too much to the translation efficiency of this linear mRNA. And this thing is still on paper. This thing yet to be developed and yet to be tested. The last one is the mutation or correction circular RNA. In this case, if someone needs to mutate circular RNA or a amino acid on the circular RNA, they can use this DCAS13 ADR2 system. It will replace, or it will helps in the deamination of the A nucleotide with the C nucleotide. However, with the mutation in this system, this thing can also be applied to this approach can also be applied to other nucleotides that are available in the RNA. It is A adenine, tyrosine, uracil, guanine. However, this thing is also on paper. This thing is yet to be established and yet to be tested, and before we can apply this for any commercial scale. I hope you got some idea about this, how the circular RNA can be modulated in the mammalian cells. If you need to go through this thing in details, if you need to go through this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.